Welcome everyone to this week's edition of the Extracellular Vesicle Club. I think this is going to be a very exciting event today because we're going to be talking about a topic that is near and dear to everybody's mind. And actually, we have a lot of these things, or we've had a lot of these things in our bodies. So if you've been vaccinated against SARS-CoV-2 in the past few years, you've probably, not all of you, but some of you would have had access or have had exposure to lipid nanoparticles. So today we're going to go to Sweden. Actually, this is the second week in a row that we've had a presentation from Sweden, but I'd like to welcome Hadi Valadi and Mohammed Nawaz, who are going to share with us their recently published work on lipid nanoparticles, how they can carry RNA, and how they can also interact with EVs, and how these two very interesting forms of RNA carrier can complement each other. So first of all, I'd like to hand it over to Hadi Valadi. I think everybody on this call probably knows Hadi and knows his name from the very famous paper that was published about a decade and a half ago now that really blew the field wide open for how EVs can deliver RNA. So Hadi, welcome. And I'd like to just ask you to introduce some of your work.
Thank you very much. Hi everyone. My name is Hadi Valadi from University of Gothenburg in Sweden. First of all, I would like to thank the organizer for the opportunity we have been given to present our work. My research is about how cells send RNA molecules to each other via EVs and how to utilize this process for therapeutic purposes. A question that we have been investigating for a while is about what happens to mRNA molecules that cells taken up via endocytosis, and what's the role of EVs in this process? For example, if therapeutic mRNA molecules such as COVID mRNA are sent to our cells via lipid nanoparticles, what happens to this mRNA and what's the role of EVs? Which Nawaz, postdoc researcher in my group, will tell you more about it. Then I handle the word to Nawaz.
Yeah, thank you, Hadi, and Witwer for this opportunity, and the whole EV Club team. As Hadi has mentioned, that we have been working with the extracellular vesicles and lipid nanoparticles, and most of us know that these lipid nanoparticles have been applied for COVID-19 mRNA vaccine by Pfizer, Moderna, and some other. So we have been studying that how these lipid nanoparticles are taken by cells and how they change the functions or molecular content of the EVs, and then how these EVs further distribute lipid nanoparticles, mRNA, to other cells, and how they are functions. So I'm going to present our recent paper that we published in Science Advances. Before I present my work, then a brief history. Most of you might know, but the people new in the field. So the Hadi Valadi first published a paper in Nature Cell Biology along with John Lottival in 2007. That they were the first time to show that the EVs transport mRNA between the cells, mRNA and microRNA. So this was a novel mechanism of genetic exchange between the cells. Based on that, this study have been reproduced by several other studies by known researchers, that James Rack in Canada and Sandra Brakefield from USA and the Roth-Cizak group also showed that this study have been reproduced by several other groups that the RNA has been. So this is the brief history. And then there was that, okay, they transport endogenous mRNA or RNA between the cells. Is it possible to insert exogenous RNA? So these were the two studies that were have shown that it's possible to insert siRNA or exogenous RNA into the exosomes. And both of these studies were the first to show the electroporation method. That has been reproduced by several other groups, by the way, that RNA can be inserted by electroporation into the exosomes. Sorry, EVs now. At that time, people used to say exosomes. And yeah, so that can be transferred to other cells. And then lipid nanoparticles have been a few decades by now been used as RNA delivery vehicles. Initially, they were used as siRNA vehicles, but later they have been tested with mRNA, and then COVID-19 mRNA vaccines we have most of us received already. So briefly about the composition of the mRNA, this is a more chemistry and polymer science. I am not good at it. It's more about a formulation thing and more physical chemistry. So basically, the lipid nanoparticles contain 4 different components. One is the key component is the ionizable lipid. That's a cationic one, and then cholesterol, the SPC helper lipid, and the PEG lipid, and then polyethylene glycol. And then there is a mRNA or siRNA this inside. So the Peter Cullis from Canada, they were and Robert Langer, they were among the leaders or pioneers in the study of lipid nanoparticles, and they have formulated and they have advanced the field of lipid nanoparticles. And these are a few structures I'm showing from different labs, and including the AstraZeneca. So our collaborator, Lennart Lindorff from AstraZeneca, they published one paper in PNAS 2018 and showing that how this orientation of mRNA is in the hexagonal phase of lipid nanoparticles inside, and how it's they have shown its stability and delivery and uptake and the translation of mRNA. And what we have been receiving from AstraZeneca, the LNPs, their loading efficiency has been very good and up to 98%. So this, and then when we talk about the RNA delivery, so it's not only mRNA, because at the end, the aim is to achieve a function inside the cell cytoplasm. So we can either deliver mRNA, siRNA, antisense oligonucleotides, or RNA editing or DNA editing CRISPR machinery. So the first thing that LNPs take this mRNA or siRNA, any oligonucleotide inside. So either they can produce a protein of interest, and in case of CRISPR, Cas9 protein is produced and it goes to the nucleus, and in the nucleus is added to the DNA, and then again, this edited transcribed mRNA, corrected mRNA goes out, whether you suppress the protein or you can correct the protein. So similarly, in case of siRNA, we can also suppress the protein, and the RNA editing, we can also edit the RNA correctly at RNA level, not at DNA level. So this is just a basic brief background. And what we have been doing, we have been studying the endocytosis of LNPs with mRNA and their fate inside the cell and endosomal escape, and those things we have been studying. So most people in the drug delivery and mRNA delivery are familiar with that. What is the biggest challenge, major key bottleneck in the field is the endosomal escape. Because when these LNPs are endocytosed, several studies have shown that within the 1 hour or 2 hours, 95% of the LNPs are inside the cell. So when they are uptaken inside the cells, and now there are enough amount that is inside the cells. But the problem is that that's not available in the cytoplasm to show the function. So hardly like 2% or 1% of the total RNA, siRNA, mRNA escapes the endosome and reach to the cytoplasm, either silence the mRNA in case of siRNA, or produce a protein if we deliver mRNA. So this was the previously thought by other researchers. So what we have connected, a missing gap, was that, is there any other aspect that we are missing? For example, when we talk about the endosomal escape, so there are the multivesicular bodies are recycling to the plasma membrane and outer membrane vesicles. So the part of mRNA could also be secreted or packaged into the exosomes, extracellular vesicles. And then we have been studying this, and we have shown first time in Nature Communication paper in 2019, we showed this mechanism that there is a linkage between the endosomal escape of the LNP-mRNA, and then it's loaded, dissociated inside the cytoplasm, and then it's loaded into the EVs. So I'm not going to present this whole mechanism, but briefly that when they are endocytosed, the LNPs that contained mRNA and the ratio between the ionizable lipid and mRNA was 1:3. But since the mRNA, the lipid nanoparticles surface charge is positive and endosomal membrane compartments are negatively charged, so it must be neutrally charged to escape the endosomes. So when we show the experimentally, it was shown that the ratio between mRNA and lipid nanoparticles in EVs was 1:1. That was a one like confirmation also at consistent with the pH change at the neutral pH. The dissociation was the highest. To just briefly tell that it was possible to detect the mRNA in EVs. If you were interested, you can see the whole the process how we figure out this. And then there was a follow-up study that what is the effect of LNPs to the EVs, like, and how these EVs respond to the LNPs mRNA, how they are changed functionally, and their endogenous mRNA and exogenous mRNA, how they respond in a bigger picture when they are transported between cells taking the LNP mRNA, but also newly transformed content in response to the LNP mRNA. So the first we used lipid nanoparticles with the VEGF mRNA, and we saw the endocytosis. And then before using mRNA, we labeled them with carbon C14 of cholesterol was radio labeled, and we checked the uptake of LNPs. So these LNPs were just without mRNA, and we just wanted to see how quickly they go inside the cells and disappear from the media. So we see that amount of lipid nanoparticles outside the cell, i.e. in the supernatants, started decreasing quickly. And at the same time, inside the cells, the carbon labeled components started increasing. So it was very aligned with the disappearance from the supernatant and amounts inside the cells. So then we further quantify, we loaded radio labeled mRNA, like sci-fi mRNA, in LNPs, and we detected them inside the cells, and we checked that the peak was around between 5 to 24 hours, and most of the LNP mRNA was detected in early time points, like peak was 5 hours. So this was just to show that LNPs are uptaken by the cells. Not only LNPs, but their mRNA was also inside the cells internalized, and the process is very quick and fast. And then we check this with the VEGF mRNA, and it's again, it aligns when the lipid nanoparticles from the supernatant started decreasing, and the mRNA inside cells start increasing quickly, and then it started in parallel, the translation of the mRNA was happening, and then it was started increasing. So it was very beautifully aligned, all the aspects, uptake and the detection of mRNA inside the cells and translation into the protein. And as we see that the first thing we wanted to see if it has been have shown the endosomal escape. If the mRNA is inside the cytoplasm, it must have been translated. So we could see the VEGF protein, and there was a huge difference between the untreated and treated. And here, one thing to mention is that since VEGF cells have VEGF protein and mRNA endogenously produced, so it was obvious that we could see a minute amount of endogenous VEGF mRNA in untreated cells. But when they were treated with LNP-mRNA, and we could see abrupt change in the amounts of mRNA inside the proteins and mRNA. And then when we studied the kinetics of mRNA first inside the cells, and we could see again, as I have shown in the previous slide, that the peak was like very quick and in early time points, and later it started maybe degradation and translation into proteins, and then we could see the decline in mRNA inside cells, and then the protein levels inside the cells started increasing. And since VEGF protein is a secretory protein, we could also see in the supernatant. So then the next was that we could see this mRNA inside the EVs. So the mRNA inside the EVs was a little bit later than the peak of mRNA inside the EVs were a bit later than what was observed in the cells. So why it's significant, why it's important, because these cells need some time to process the mRNA, endosomal escape, go to the endolysosomal system in the cytoplasm, and then package into the exosome. So this again further confirms that first the LNP-mRNA is inside the cells, and then later it starts secreting into the EVs. So we use the different cell lines here, like HTB is a lung epithelial cell line, HOVIC and CPCs are cardiac progenitor cells. So we also tested in different cell lines that, and we could see, although the amounts were a bit variable, but it was possible to see the mRNA was secreted into the EVs from different cell lines. And the one interesting thing was that although untreated cells had, like here we could see a minute amount of endogenous mRNA, but their EVs could not have like this. So it seems that the process of packaging is very, very, very selective into the EVs. And then we characterize EVs that whether there might be some contamination, that if EVs have been interacted with the LNPs outside the cells in the supernatant, and we fetch these EVs with the antibody, CD63 antibody, conjugated beads, and then we and these EVs contained also Cy5 mRNA, and then when we conjugated them and we washed them and we analyzed with the flow cytometry, and we could see at different time points the EVs that were positive for CD63, they were also positive for Cy5 mRNA of LNPs. Here again, the peak was around like 5 hours, as consistent with the VEGF mRNA, that was 2 to 5 hours was the peak. And as a control, we used LNPs and beads only to see that maybe these beads, LNPs can bind with the beads as a artifact, and then maybe our EVs were not conjugated only along with the CD63, but maybe LNPs. So we use this LNPs that contain Cy5 mRNA with these CD63 beads, and we could not see that. So this was just a control to show that, EVs contain this LNP-mRNA. And further, since several studies, the people who work with these lipid nanoparticle fields, they know, they are familiar that the uptake occurs very quickly and in early time points. So in our case, we saw the peak was before 2 hours, and maximum at 1 hour, most of the LNPs were inside. So what we did, we washed the cells after 1 hour, and we added, after 1 hour of LNP treatment, we washed the cells, and then we added a fresh media that didn't contain any LNPs. And after waiting 24 hours, we collected fresh EVs, and we could still find that there is a mRNA, VEGF mRNA. What that means that this mRNA in newly secreted EVs is coming from the cells that those cells already have uptaken the LNP mRNA before the wash. And when we compare the VEGF protein in the inside the cell lysates and its supernatants after washing and before washing, and there were no big differences in the protein production. What it means that the most of the VEGF mRNA is also already inside the cell before washing. Otherwise, there could have been a significant difference. And the same, the total RNA and total protein content didn't change so much after the wash. And so this was the one confirmatory experiment that the LNPs were already taken up by the cells, and then they are secreted inside in the supernatant, where there's no LNP left to interact with EVs to show that the factor of contamination. And then we also analyzed the presence of CD9 in different EVs from different cell lines, and they were positive for. And to us, CPCs were like positive for CD63, but they were not positive for CD9, because these EVs first were captured with CD63Bs, and then they were further captured with CD9. So they already are positive for CD63. But one of the cell lines, EVs from cardiac progenitor cells, didn't. Either we are not sure, either they don't have CD63 or they don't express. So we are not sure about that. And then other than the mRNA, we were also interested to know that whether, because we are delivering mRNA via EVs to other cells, and whether the EVs already contain VEGF protein. And when to us, the good method was to use the size exclusion chromatography, because in early fractions we have the EVs, and in later fractions we have the free proteins. And then we see that in early fractions we could not see much VEGF protein, free protein, or in EVs inside the EVs, but it was a free protein that we detect in later fractions. And most of the mRNA was in early fractions that are the EVs. And then we also analyzed the size for zeta size, and that it was around 100. And then we also have in this publication nanosite, and then I'm not showing here, but the size was confirmed. And also we did morphology. Yeah, that's not shown here. And then the details that we did we went a bit further to study their stoichiochemistry and the copy numbers when we deliver LNPs into cells, and how much mRNA copy numbers and protein copy numbers can be detected inside the cells, and how much we can detect in the cells inside the EVs. We delivered 3 microgram of VEGF mRNA via lipid nanoparticles. The number of particles we quantified were the 2.5 raised to 10 raised to 11, and the average number of VEGF mRNA per LNP particle was 26. I mean, one lipid nanoparticle contains around 26 mRNA copies, and then number of VEGF protein copies transferred to cells. So this is what we transferred. So we know that how many LNPs we have in a certain volume and how many microgram we have. And in the publication, there is a method we have shown that how we can calculate these copy numbers. So this is the first table shows that how much we have delivered. So after delivering 24-hour, this table shows after 24 hours of delivery, how much we could detect inside the cells. So we could see the average number of protein copies in the cell lysates is 2.4. I mean, if we compare how much we deliver and how much we get, it's really, really negligible. It's too little. So the average number of VEGF proteins is 0.1. What it means that if we deliver 1 mRNA molecules, it produced 0.15 copies of VEGF protein. What happens to the rest of the mRNA? I mean, it's 99.99% is not there in the cells detected, and it aligns with the previous studies that shown it's the part that is detected inside the cytoplasm is too little. Either it has been degraded by the lysosomal system, and either it has not been escaped at the time we harvested the cells, or it has been secreted into the EVs at that certain time points. So when we see that, when we compare the copy numbers inside the cells and EVs, we could see that at early time points, there were less copies inside the, at that time point, I mean, half hour, it might have not been escaped. But at 1 hour, we see the peak again. And that aligns with our previous, in previous data I have shown. It aligns with that the peak is around 1 hour inside the cell. And in EVs, it's around 5 hours. And we see that only 31 copies were detected at the peak. The maximum we could detect in EVs were 31 copies of VEGF mRNA out of 2.5 10 raised to power 11 copies we delivered. So if we see that trillions of copies were delivered and we could only detect 31 maximum in one, and then at peak, but then in other time points were less. And then it shows that the first LNPs are metabolized inside the cells and they are packaged into the EVs, but still we have been receiving some criticism that it might have been contamination. But if it was contamination in the EVs outside the cells, we don't know how much it is, but if it was a significant contamination, we should expect hundreds, at least thousands of copies in EVs, as we have delivered trillions of copies or billions of copies inside the cells. If they were not uptaken by the cells, if they were contaminating the EVs outside the cells, so we could have detected. So these are the clues that tell us that the EVs are taking their LNPs mRNA. And then further that we thought that we can deliver these EVs to the other cells that contain mRNA from the LNPs, and we see that whether they are functional. And as we have already confirmed that they contain Cy5 mRNA, and when we deliver to the recipient cells, and we could see that not only the mRNA was inside cells, but also we could see the translation into the EGFP, enhanced green fluorescent protein. But what was our hypothesis, and if that was supported by the results, was that when the LNPs are uptaken inside the cells, as part of its degraded, part of it goes to the escape and translated, and part of it, because it's a secretory protein of VEGF secretory protein, it can be secreted. But part of it goes to the EVs, and it can be distributed to the other cells in the certain population in the neighboring cells. And what clinical relevant here is that, as Witwer Ken has introduced in the beginning, that it's relevant with the COVID-19 vaccine, mRNA vaccine, that many people have receiving it. So if this mechanism is working in a way that EVs take up the LNPs mRNA, and then they secrete it and they transport to the other cells, and then it might happen in our body. Although we have not studied this, but it's quite possible that since EVs are uptaking the LNPs mRNA and transporting between the cells, it can happen. But at least we have shown in vivo studies it was possible. So as we were interested in to see that how these LNPs change the mRNA, or in general the content of the EVs, because the VEGF is an angiogenic molecule, and it's involved in the vessel formation. And when the LNPs were delivered to cells containing VEGF mRNA, and the cells have shown response to the LNPs, and this response was reflected inside the EVs, because these EVs not only contained VEGF mRNA, but also several other angiogenic mRNA transcripts were upregulated and secreted into the EVs. So we have shown here a certain set of mRNA, angiogenic mRNA here, but there are several other transcripts that were selectively packaged into the EVs of LNP treated cells versus that were not treated with LNPs. So it means that the LNPs of this VEGF mRNA, or it can be true with other mRNA that's function A versus function B, that can be reflected into the EVs through the cellular routes. And then we further transfer these EVs to the recipient cells, and we could see they were enhanced proliferation and a number of nuclei size, and this reflected that there is a proliferation happening. And then we were interested to see, we did a pilot study to see that whether it's a vessel formation and network. So we saw an initial clue that there is a difference between the cells that were not treated with VEGF mRNA by EVs versus those that were treated with EVs containing VEGF mRNA. And then we further analyzed the networking analysis, in using different EVs. And here it was very interesting thing to see that since LNPs, we see in all of the in the panels, we could see this red panel, you see these represent the untreated, and then if we see the LNPs, so if the LNPs were showing the function itself, the mRNA of LNPs is translated into the VEGF protein, and if it shows the angiogenic, it should be expected more than the EVs. But for us, interesting thing was that the CPC EVs, they produced less protein when we delivered equal amount of VEGF mRNA. Here 60 nanogram were delivered via LNPs or lung epithelial cell line EVs or HOVEC EVs or cardiac progenitor EVs. So despite we delivered equal amount of mRNA, the amount of protein produced against each EV type was different. But not only this, if we see here in panel G, the extent to which the network formation was observed was highest in case of CPCs per VEGF protein produced. They produced less protein, but they were highest. They are very efficient in producing the networking formation. So here, the point is that these LNPs contain only one mRNA, but these EVs contain several other angiogenic RNA. And also the cardiac progenitor cells show that because they are they're very clinically relevant, because in the cardiac tissue, we need to produce angiogenesis. And if the CPCs are showing a higher networking, and it could be very interesting to show in future studies. Here, to summarize that LNPs are extending their functions through EVs, or in other words, EVs are extending the functions of LNPs, but not only this, but they have been transformed with other additional molecules that were not in LNPs themselves, but they have acquired. And then we directly injected the VEGF mRNA into the heart to see that whether it can be uptaken by the tissue and it can be translated. The reason we directly injected here is that AstraZeneca has published the clinical trials in the patients going through the heart surgery, and they injected directly to the heart by opening the chest and then inject into the heart. And then they have seen that the clinical trial have shown that there was significant improvements in the function and vessel formation. And we just try to recapitulate the same thing. And here we just simply wanted to show that whether the direct injection to the heart can produce VEGF protein when we inject VEGF mRNA via EVs or LNPs. And we could see here that different EVs could produce VEGF protein into the heart. And then we also collected the non-injected area of the heart, and we could not see the significant differences between the in the non-injected. What it means that it stays in the local area, it isn't diffused to the other area. So where we want, we can target the targeted area, we can produce the protein where we need it. And it did not leak into the blood or liver. So there were no spillover to the other organs. And this is, again, is very important because VEGF is a carcinogenous protein. In case of a tumor, it facilitates the tumor growth. So it's not, and maybe other organs, it's not good to have to produce extra angiogenic proteins. So it was very interesting to see that it's not spreading into the other, especially in the liver, because most of when we see via IV delivery of luciferase mRNA, and most of the mRNA was observed into the inside the liver. And this is consistent with other studies, that several studies have shown that mRNA or other drug modalities, they are accumulated in the liver. So, but in case of direct injection of VEGF to the heart, we couldn't see it spill over to the liver. And then just we wanted to see that whether there is no interference of the results, that if there was some endogenous VEGF mRNA or protein and we are delivering to the other, and what we are achieving the results is not due to the EVs, it's due to the endogenous. They already, it's not due to the VEGF mRNA of LNPs, but maybe it's due to the endogenous. So when we did this experiment, we could see that although there were a minor amount of endogenous mRNA inside the EVs, but when we deliver to the recipient cells, we could see that they were in the untreated cells. We could not see the VEGF protein. The mostly we observed inside the treated ones. So this was just a control. So summarizing the work, that was like when the cells uptake the LNP-mRNA, so they respond to. So in this case, this was VEGF mRNA. So cell responded to the VEGF. So VEGF might have activated the VEGF or receptors or some other pathways, and those, in response to the VEGF, several other transcripts were activated and overexpressed, and those transcripts were secreted inside the EVs, and they were further distributed to the other cells. And a part of that was secreted out. Part of the translated was secreted, as VEGF is a secretory protein. So yeah, this is the study that we recently published in the journal Science, Advanced Science. Yeah, sorry, sometimes I confuse with the Science Advances and Advanced Science. So there are two different journals. So it's Advanced Science. At the end, I would like to acknowledge all our collaborators and the work. It has been very fun working at Hadi Valadi Lab over the almost now 8 years. And initially, I have been working with the postdoc Marco Maggiore. We published in Nature Communication together a method to load a mRNA into the EVs. And then with several collaborators from the AstraZeneca and from University of Kobe, and of course, all the authors who have been helping us to accomplish this study. And then, of course, the funding agencies, Vinnova and the Swedish Strategic Research Foundation. And this work was also within the one consortium, Formula X, that is between the different industries and the universities within Sweden. And it was conducted within that consortium. And of course, at the end, my family have been very supportive to me during all these times that I have been working. And it was always fun to come home and see kids. And at the end, if you have some questions, and you're welcome to ask the questions.
All right. Well, thank you very much, Nawaz, for presenting this work and for doing such a careful study. I have now allowed, I've allowed people to unmute themselves and to start their videos if they would like to. So we will now have our question and answer period. I see that we have, let's see, some people have sent something to me, and we have at least one question in the chat box already. So just as a reminder, if you do have questions, comments for Nawaz or Hadi, please put those in the chat box, and then I will get to you in order. So let's go ahead and start now. And we will start with Anand. Anand, you have a question about cell types.
Yes, thank you, Ken. Nawaz is a beautiful study. For some reason, now I'm there. I'm sorry. Yeah, quick question. I might have missed it while you said this. So did you try harvesting EVs from LNP transfected cells, say from a mature cell, like a muscle or a neuron, and putting it back to a embryonic cell type where there is no differentiated state, and ask them to differentiate? Have you tried something of that sort?
We have not tried that, but what we have done, like we treated one type of cells with the LNPs, transfected or treated. So we harvested the EVs and then we delivered to other type of cells. For example, from lung epithelial to the HUVEC or to the CPCs, or from CPC to the HUVEC. So we have done that type of study, but not to the neurons or differentiation. That we have not done. But what we could see that they from one cell type to the cell type, the message is here that EVs transport between different cell types, transport the LNP-mRNA between different cell types.
One continuation of the same question is like, all the cell types, what you've actually mentioned in the study, are cell lines, and none of them are actually primary cells, right?
Yeah, except for the lung epithelial that is immortalized. Other ones are the yeah.
But still, they are like example.
Yeah.
So you're just coming back to your summary where you said that like it has the potential to deliver to one cell type, and they can give it to the other cell type. I think if you want to answer that, for me, it would have been more interesting to have you tried on any primary cell cultures rather than cell line, which has the ability, which won't survive for a long time, but still can actually, these can do something to them or not. And another thing I would like to add is that have you tried looking into lipid vesicles? You said you transfect cells with mRNA, and then you won't actually find a lot of them coming into the EV. But there are lipid vesicles inside the cells, which have also been budded by other cell organs. Have you tried looking into those fractions?
Yeah. So if I understood your question, that whether the content of lipid nanoparticles is also in the EVs, that's what I understood.
Also the EVs or in lipid vesicles in the cells. The cells makes other kind of lipid vesicles, not exactly EVs, but like budding from other like Golgi or ER or whatever. Then they're still like a vesicle.
No, we have not.
Okay.
No, we have not studied those. But what we have seen that in our Nature Communication paper that the ionizable lipid part of ionizable lipid was also inside the EVs. So it means, but if they are making some other artificial vesicles other than EVs, so that we have not studied. So.
No, perfect. Thank you. It's such a beautiful, well-calculated study. Thank you.
Hadi?
I would like actually to comment also to your question. It was very good questions. No, we haven't looked at the differentiation of the stem cells to something else. But we have delivered them in vivo, either by direct injection or by intravenous injection. And we see that actually in vivo, the EVs or LNPs can deliver the mRNA to a number of different tissues, and the mRNA is functional and can be translated to a protein that mice lacking. So we deliver the mRNA coding for a human protein. Mice lack this protein. So if they produce it, so it must come from the mRNA. So both the EVs and the LNPs could deliver the mRNA, and the mRNA was functional and could give rise to a new protein that mice was lacking. But Simon says, no, they haven't looked at the differentiation. No, that was a good idea.
Yeah, I think if in vivo works, that's brilliant. That's a beautiful thing to show, actually. And I might have missed out on the whole topic because so much of data for me, trust me, it was beautiful work. And thank you for answering all the questions.
Welcome.
Thanks, Anand. I have a question that has to do with something that you did raise briefly in the paper, and that is about the immunogenicity and what the implications are here. So how, as you develop these associations and this technology, how can you maximize the opportunities, I guess, to give repeated treatments to the patient by avoiding immunogenicity? What do you think are the next steps in making sure that you can use this EV LNP interaction to do that?
Hadi,
I have one. I can actually just comment. Nawaz, you can continue afterwards. So mRNA delivery to human, we know that actually viruses has been used, lipid nanoparticles has been used. The problem with the viruses, all of us, we know that actually is the repeat, as you said, is the repeat treatment. So the body actually starts to react and because of the our immune system. The good thing with the lipid nanoparticles is that it's possible. I mean, all of us, we get several doses of lipid nanoparticles containing COVID mRNA. And that our body or our immune system cannot react that way, like viruses, to lipid nanoparticles. Then we come to the EVs. I think actually we never administered several doses of EVs. We have done only one dose. So we cannot exactly know actually if the EVs work as a virus or lipid nanoparticles. I don't know, am I sure that we haven't treated several?
No, we didn't do multiple treatments. In vitro, we have done in some experiments to see the repeated
exactly.
time-lapsed. But in what we could see that good thing with EVs, at least in 2 papers, we have shown that EVs provoke less immune response compared to LNPs. Because the problem with LNPs, they have the PEG, polyethylene glycol, DPC, cholesterol that are additional to the cells that cells don't like it. But EVs don't have those things. So that can be one advantage in future. That's what I think that.
I like the question. I really like the question. I think it's worth to test it to see if EVs with several different treatments can act like a virus or lipid nanoparticles. Thank you for the question.
Yeah, I do also think that it's important, and there's even the possibility, if you have a therapy that's based upon mRNA LNP treatment, and you're worried about this, that there could actually be a contribution of EVs from the patient. Lots of ways that could be done. To sort of cloak a treatment with a native EV, so to speak. So lots of opportunities there. Yeah. I have a question that has come. Oh, go ahead before we move on.
I mean, it was the same concept that you say that autologous or allogenic, I mean, take from the same patient or host and give back to that. So it will be considered as a, homing. So,
Yeah. So the other question that I had that came in to my box, I don't think it's here in the chat box though. That is about other types of RNA. Do you think that there's also an opportunity to use LNPs or LNP-EV combinations for delivering other RNAs besides messenger RNAs?
CRISPR has been, like some study, people have shown CRISPR, siRNA, and one that I saw one there, like a group, they have combined, like collectively incorporated siRNA, mRNA, and they delivered. It was possible to, initially the LNPs have been used for siRNA, but if we combine EVs and LNPs with different, we have not tested at least siRNA and EVs, So we have been working, but other people have shown CRISPR or other siRNA.
I can also actually add something to this. So is that we have tested mRNAs from around 500 nucleotides to mRNAs up to 1,000 nucleotides. So, and all of them, it was working that we could find part of this mRNA inside the EVs. So if I'm guessing, we haven't tested other type of the RNAs, siRNA, small non-coding RNA of any type. We haven't tested. But if I'm guessing is that actually a small RNA is no problem. But the question is, if we go with very big mRNA, like 4,000, 5,000 nucleotides, does it work? Because our research shows that the mRNA needed to be neutrally charged to escape from the endosomes. Such a big mRNA. So it would be actually very interesting to test it. So we don't know.
Yeah, so maybe the need for there's very specific, packaging mechanisms that viruses have evolved to get those very long RNAs into the particle.
Yeah.
Good point. Good point. Okay, and then the other question that I got was surprise about the EV corona or the corona of the nanoparticle. So how have you looked at or have you thought about how this association, this accretion of factors from the host organism might differ in interacting with the LNP versus the mixture?
We have not studied this, but it's like quite possible, especially when you see the samples from the animals, like in blood samples or something. So there are the more chances to my knowledge compared to the in vitro studies, because those have a lot of other proteins, high density lipoprotein, or that can make a protein corona. But one of our within this consortium of Formula X study from the colleagues from AstraZeneca and Chalmers, they have published something about corona that with LNPs, they treated the cells with LNPs, and then they said that what happens to the it was not EV part, but that was part of LNPs itself gets something that way related to this question corona. But I mean it's quite possible to, but we have not studied that.
Very good. All right. Well, it looks like we have come to the end of the questions that I've received anyway. So unless somebody else has something pressing that they'd like to interject here at the last minute, I'd like to thank both Hadi and Nawaz for sharing this work, for doing the work in the first place, obviously, and then for sharing it with us today in EV Club. Gives us a lot of good insights and questions that we can continue working on. So all the best to both of you, and thanks to everybody who has joined today. Hope you have a great rest of your week, and we look forward to seeing you again on EV Club in the near future. All right, take care, everybody.
Thank you, Ken. Thank you. Bye.
Bye now.